Everything is personal right here. Everything is personal right here.
Ladies and gentlemen, boys and girls, welcome to another episode of Everything is Personal. And today we have a really special guest with us, Dr. Henry Erlich. Thank you for being on.
Oh, thanks, Len. It's a pleasure to be with you.
You're such a pioneer in the industry. I really want to get to your personal sort of story of how you got started, but I'm interested in what sort of drew you to the field of genetics and molecular biology in the first place? Was there like a moment that said, oh, you know what? This is kind of where I want to pursue?
Yeah, no, there's definitely a moment that I can define as taking a course my freshman year in college. I had to pick a major as a freshman, so I picked Renaissance history and literature.
It's close to genetics.
Yeah, it's very close. It's an easy transition. So anyway, I took an introductory course in biology taught by this brilliant lecturer, George Wald, who later got the Nobel Prize for his work on the biochemistry of vision. But anyway, he was a really inspiring teacher. And so it made me basically I shifted my major to biochemistry and I took a genetics course. And it was really the sort of conceptual elegance of genetics that led me into molecular biology.
So let's go into your origin story a little bit. And I want to dive back into and unpack that a little bit more because I know, we talk about DNA and we talk about genetics. And you and I talked right before this, like, what do people know? What do people don't know? I'm always shocked when having discussions with healthcare professionals that really don't understand genetics, and they probably just got their DNA 101 course in school, and that's it. They never had to deal with it in in their professional journeys, I guess. But before I get to that, where did you grow up?
Well, I was born in New York. My parents were immigrants too, and then from Poland. They escaped during the war, and then when I was born, my dad was actually in the U.S. Army because he enrolled as soon as they moved to the United States. But I think when I was about 3, my family moved from New York to Seattle, which is where I grew up.
Got it.
And I went to a public high school there. And I actually, in high school, I didn't take any biology courses. I took physics and chemistry and, a lot of the other things. But I actually started teaching myself biology because I thought it was fascinating. And then when I went to college, I majored in, as I said, Renaissance history and literature, but I took this totally wonderful introductory biology course. And then, as I mentioned, switched to study genetics and biochemistry. And then I went on to get my graduate degree in genetics, but I went back to the University of Washington because I love Seattle, where I had grown up, and it had a great genetics department.
When so you're living in New York, and as a kid you moved to Seattle, was that for you for work? And what was the family dynamic? Were your parents together? Do you have siblings?
Yeah, I do have a sibling who was born in Seattle, but my dad was studying Russian literature at Columbia University, and his first job was at the University of Washington in Seattle. So that's where the family moved to Seattle. I was about 3, and that's where I grew up. I went to Lincoln High School there, and actually, as it turned out, after I graduated from college at Harvard, I went back to Seattle to get my PhD at the University of Washington because I really like Seattle as a place to live.
You like the weather?
No, people put up with the weather for the mountains.
Yeah, that's true. I spent a little bit of time in Seattle. I was doing some consulting work, not really Seattle, but with Microsoft. But,
right. Sure.
But prior to that, I was, or I guess, before that, I was in the music business, so to speak. I was working with Tower Records for Tower
Mhm.
Records
Oh,
as
right.
a buyer. So there was a lot of things that were going on in the Seattle music scene at the time. So I could include myself in the quote unquote grunge scene. So
Yeah.
that
Yeah.
was interesting.
Seattle, yeah, does have a very lively musical history and scene. I remember when I was a kid, a teenager, I used to go to Ray Charles concerts
Uh,
because, he was traveling around the world, but he started out in Seattle well, he moved from Florida to Seattle as a teenager, and as you may know, worked with Quincy Jones there.
oh yeah, I definitely know. It's one a huge loss for the music industry. Quincy's passing
Yeah,
is
yeah.
so. I'm just curious because I sort of put myself in this role where you have people like Slash, the musician, that said, when I picked up a guitar when I was, I don't know, 8 years old, whatever, and I started playing, like I knew that I was going to do this for the rest of my life. Like this is what I connected to. As a kid, did you have any aspirations of, I'm going to be a scientist, I'm going to be a doctor, or like what kind of kid were you? Were you into math? Were you into sports, music?
Well, I was into sports and music. I played basketball and I studied classical guitar. But I was interested in a lot of things in literature, philosophy, politics. And it's true, I was very taken with the rigor, the intellectual rigor of science. So as I mentioned, I studied physics and chemistry, and I read a lot about biology, but I never actually took a biology course in high school. Um, but so I wasn't really sure where I wanted to go. And when I had this kind of revelatory biology course, I took it. I majored in biochemistry and genetics, not so much because I thought this was a career choice, but because it was a very interesting field to think about, and literature and things like that. I would say kind of came easy to me. My dad was a professor of literature, and I've maintained my interest in literature and poetry writing and stuff like that. But science was a new adventure for me. And then obviously, as it turned out, it became a career choice.
Yeah, it definitely when I think when you have a real interest in something, you sort of pulled by that as a vehicle instead of pushing through and saying, because my parents are like, yeah, I went to physical therapy school. Like, you have to be a physical therapist. We're immigrants. We immigrated. My grandmother, my dad's side of the family on his mom's side is from Poland,
Mhm.
as yours, and I was born in Lithuania, so it used to
Mhm.
be it's right next door anyway. And we're immigrants, you have this, hunger for success, but also for your kids to have some stability in their life.
Mhm.
You're going to go to physical therapy school instead of music, what I wanted to do,
Mhm.
because what career are you going to have in music? And then it's going to give you a stable job and you're going to do this and you didn't do that. So, having something that pulls you towards a purpose, you're going to find it eventually down the line. I think, even if you don't step into it right away. And you sounds like you had this interest. I'm curious, it's not just molecular biology, but genetics itself. What was so interesting about genetics for you when you started learning about it?
Well, one of the really fascinating things about genetics was so much could be discovered or inferred from extremely simple experiments. So, for instance, two of my heroes were Francois Jacob and Jacques Monod, who were French molecular biologists. They work with bacteria, and they basically were the first people to figure out how genes were regulated, how they were turned on and turned off by simply studying different mutants of the bacterium E. coli, so that they were able to conclude very profound things about the world and biology by just looking at different mutant strains of E. coli and how they responded to environmental conditions, or the fundamental rules of genetics known as kind of Mendelian inheritance from Gregor Mendel. the abbot in Brno, what's now, I think, Slovakia,
Mm.
he figured out how genes were transmitted by simply looking at round and wrinkled peas and counting them. And he inferred all of the basic rules of inheritance simply by these simple observations. So I think genetics was interesting to me because you could have such profound insights from sort of simple, elegant experiments.
I find it interesting, and I would like you to elaborate on this because, you know, people who don't have scientific knowledge, like your average person, they talk about, I just have, it's in my genes. I genetics. I got it from my father. I got it from my mother, and I was always fascinated. I'm like, well, just because you have a genetic predisposition to something, it doesn't mean that you're in jail. You're not in genetic jail. You have it's a godsend. It's a blessing for you to know this so you can take specific action to be able to, as you said, regulate the expression, epigenetically. So I was wondering, maybe you can talk to people a little bit about what I'm referring to when people have their genetic predispositions or they know what their genome's like. What can they do? Does that mean that they're sort of destined for this, or they can take action to be able to regulate the expression of their genes?
Well, it depends. There are a lot of different genetic systems. Some are what are called monogenic diseases. There's a mutation in one of your genes. So, for instance, I've done a lot of work on sickle cell anemia and beta thalassemia.
Mhm.
So those are all mutations in the gene called beta globin, which ends up being part of the hemoglobin that transports oxygen in the blood. So if an individual inherited a mutant gene from the father and from the mother, then they have two copies of this mutant gene. And, there's a very, very high probability that they will get sickle cell anemia or beta thalassemia. Now, there are things that can be done. Um, for beta thalassemia, you can have transfusions, you can have bone marrow stem cell transplants. And as you may have heard, Len, that there's now gene therapy protocols for sickle cell anemia. So there are things that you can be done for those diseases where the genes are highly predictive. But as you pointed out, there are other kind of genetic predispositions. Like I spent a lot of my career working on genetic predisposition to type 1 diabetes, which is an autoimmune disease. So there's some genetic profiles where you might have, a 1 in 50 chance in getting type 1 diabetes instead of a 1 in 300. So, you have an increased risk, but you're, the chances are that you're not going to get it at all. And you can be screened for it. That before you actually get clinical type 1 diabetes, you develop what are called autoantibodies. And so that can be detected. Now, fortunately, with the advent of insulin, type 1 diabetes is a totally manageable disease. Now, there's another type of diabetes, type 2 diabetes, which has another set of genes. And the predisposition isn't quite as great, but there are genes that predispose you to type 2 diabetes. But as you know, there are lots of environmental or lifestyle steps that you can do. You can lose weight, exercise, and then there are also medications that help manage type 2 diabetes. So genetics should never be viewed as a death sentence or something that's immutable.
Yeah.
There are usually interventions that can be made.
Yeah, one great example you used was sickle cell because I was listening to a talk. I went to see Peter Diamandis talk about CRISPR, and he was talking about we have the ability today to be able to address these genetic disorders before they manifest themselves. So you can go in and, replace an affected, gene before it's expressed. And what he was saying was that there's a lot of political related pressure that prevents them from doing that, I guess in the womb or from an embryonic state. So I was just wondering, technology is so fascinating, and I want to get into like the PCR stuff as well, but I'm curious about your feeling of how machine learning and AI, how they can actually utilize that right now to start addressing some of these conditions before they show up. Because one of the things you brought up, diabetes, for instance, one of the things in diabetes is, yes, you have a predisposition, but if, instead of, reducing the cost of insulin, instead of managing it, maybe there's a way that we can predict and prevent some of these conditions to come up in the first place. So the question is the use of machine learning and AI, and seeing if there's a way to use technology like CRISPR to be able to address some of those conditions before they actually express themselves.
Well, as I said, type 2 diabetes, has to do with it's a metabolic disease. It's not an autoimmune disease. Type 1 is autoimmune. And so there are things that can modify your metabolism. For type 2. For type 1 diabetes, it's an autoimmune disease. And the reason that type 1 used to be called insulin dependent is because your immune system attacks all of the insulin producing cells in your islets, in what's called the islets of Langerhans. And so you can no longer produce your own insulin, and you're dependent on what's called exogenous or medical insulin. But if there were a way to modify the autoimmune attack on the cells that produce insulin, you might be able to preserve your ability to make insulin and never become dependent on medical insulin. So, I think that there's actually a lot of research going on in how to basically modulate an autoimmune response, or sort of, turn it off so that you never actually become dependent on taking insulin.
Yeah, and I think, there's a lot of conditions that are autoimmune related where, our own immune system is really over expressing it or attacking us in the first place. So there's a way that we can know that up front. And from my standpoint, our focus initially was the endocannabinoid system. So modulating the endocannabinoid system and the response of the endocannabinoid system as a modulating system that is associated with homeostasis. So, we've seen a lot of things in different studies, that an overactive immune response to, cortisol expression to, having extra anandamide level that's being produced, and having a genetic predisposition to something along like fatty acid amide hydrolase, the Fa gene expression, which actually metabolizes anandamide much more rapidly based on that genotype. Now you have an overactive immune response, and people are walking around with pain and inflammation, like they're going to a doctor, like my knee hurts. Oh, we can give you a shot for that. But why does the knee hurt? That conversation doesn't enter. It's like, did you hurt your knee? I don't remember. Well, maybe you slept bad. We have a solution. We'll just shoot it up and it's going to take away the pain. But for me, the pain is always the indicator. Like, why are you shutting down the pain?
Yeah, it's the canary in the coal mine.
Exactly. So I find it fascinating. And going back to AI, I think maybe by having something that can help us identify and ask those questions up front, maybe it'll help healthcare professionals deal with the root cause instead of addressing the symptom.
Yeah. No, that's true. I mean, one of the reasons that I was so interested in type 1 diabetes is because it's an autoimmune disease. And I was very interested in immunology. It was kind of a mysterious system. And I did a postdoc postdoctoral fellowship at Stanford in immunology. And so one of the first things that we did when we developed PCR was start analyzing genes involved in the immune system. They're known as the HLA genes, and they're the technical term is the most polymorphic genes in the human genome. What that means is they're most variable, that there's the most genetic diversity at these genes. So there are possibly 1,000 or more different variants at these genes. And presumably the reason that there's so much variation, genetic polymorphism at these genes, is because they confer the ability to respond to different infectious disease pathogens. So the more variants there are in a population, the more likely that population is to be able to make immune responses to pathogens. And so once we developed PCR and I developed a system to analyze these genes, and I developed that for reasons of medical genetics, not forensics, but that was the gene that we used to do the first DNA case in the United States in 1986 because it was polymorphic. So you could get a genetic profile from the evidence sample and then compare it to see it was the same profile as the suspect.
You jumped right into it. It's exactly what I want to talk about next, because when you're developing something that's so revolutionary and you had such a pivotal role in advancing genetics in the world, based on the research that I did and the what's written about you and your own publications as well. I was wondering, when you were thinking about it, and maybe you can explain what PCR is to people as well. When you're coming up with this, did you know that it was going to play this pivotal role in genetics? And then we'll get into forensics, because that my next follow-up question was, did you even think about that? Oh, this can be utilized here.
Well, to be honest, I've always been interested in the genetics and the criminal justice system, because it seemed like a practical thing that, I sometimes use this phrase called genetics for justice, that something so basic that I work on, molecular biology and genetics, could be used to exonerate wrongfully convicted people and to convict people who've done terrible things. I always thought that would be interesting, but the only way I really got into it was because at the building where our lab was at CETUS, this was a little biotech company in the San Francisco Bay Area in the mid-'80s, there was also a forensic scientist. But I should step back and maybe explain what PCR is.
Yeah.
So I had mentioned earlier, Len, that we were interested in sickle cell anemia and studying mutations in the beta globin gene. And my colleague, Kary Mullis, who later, as you may know, got the Nobel Prize for this idea, he had this idea that in a test tube, you could synthesize millions and millions of copies of a specific sequence. So Kary and I and some of our other colleagues thought, okay, this is an interesting idea. Can we actually get it to work? So Kary and his technician went off and tried to do something, didn't quite work out. And then I was working with another group, and we decided to see if we could amplify the beta globin gene from the human genome. So it was very ambitious. That would be the test. If we could do this, this could actually be very important. But when we first started out, well, first I should explain that PCR depends on targeting a specific part of the human genome. In our case, it was beta globin, and then using the same system that cells replicate their DNA to synthesize millions and millions of copies of a specific sequence. So we used an enzyme known as DNA polymerase, which basically copies DNA and helps the DNA replicate. So the basic idea of PCR, it's actually a simple idea. You take the double helix, and as you and your viewers, probably know that DNA is a double helix, but you can separate the two strands, and you can do that by heating a solution of DNA. The two strands of the double helix separate, and then you can use one strand to make a copy, and then another strand to make a copy of that strand. And so in one cycle like that, you double the amount of DNA that you started out with. So in 20 cycles, because this is an exponential reaction, you can make 1 million copies of a sequence that you targeted in the first cycle. So that's basically PCR. And as I said, we started out trying to amplify beta globin so that we could do prenatal diagnosis for sickle cell anemia and then beta thalassemia, another disease of mutations in the beta globin gene. But after we figured out a way to do this, I wanted to turn my attention to the immune system because that was my own personal area of research and fascination, really. So we picked a gene, one of the so-called HLA genes that's involved in the immune system. And because we were able to amplify it and because it was polymorphic, when the time came, we could use it to address a forensics case. And when I say when the time came, my colleague Ed Blake, who's a forensic scientist with a little tiny lab in the same building, we would chat. And at one point he said, "Henry, I've got this problem. I have a sample that's been stored in formaldehyde. So all the DNA is fragmented into tiny bits. The proteins are denatured. I can't do all the tests that I'm used to doing, like blood typing or analyzing proteins. Do you have any idea?" And I said, well, we've always been thinking of using DNA and genetics for forensics. So I said we could try to use what was then a kind of standard technique. This was pre-PCR. And because the DNA was so degraded and present in tiny little bits, that approach didn't work at all. So I said, well, we have this totally new thing that we're trying called PCR. Let's see if we can amplify this polymorphic HLA gene from this autopsy sample. So the very first time we tried it, it actually didn't work. We were then using a DNA polymerase from the lab bacterium E. coli. And there were many issues with that. It wasn't very specific. It amplified a little bit, but it wasn't 100% specific. So then we tried a new DNA polymerase from one of the bacteria that grew in the Yellowstone hot springs called Thermus aquaticus. And the enzyme now has the name TAC polymerase, TAQ, after Thermus aquaticus. And actually, you may remember Pac-Man.
Yeah, it's TACMan. I mean, we use TACMan probes in some of our works.
Oh, okay.
Yeah, I'm
so
very familiar.
Randy Saiki, who was a technician I was working with, has a very well-developed sense of humor. And he developed something called TACMan after the video game Pac-Man.
That's so funny.
And it was because the enzyme chews things up, just like the little Pac-Man in the video game. But anyway, once we introduced the TAC polymerase into the system, PCR was transformed. It was more specific, it was more efficient, and it could be automatable. So then when we looked at Ed Blake's autopsy sample, we were able to amplify this HLA gene and use it. And that became the first DNA case in the world in the criminal justice system. And a couple of years later, we did what was personally very satisfying to me, the first DNA exoneration. It was the Gary Dotson case, who had been convicted of rape and had languished in jail for about 10 or 11 years. And we were able to show that the semen stain was not from him. So he was exonerated. And as you may know, the Innocence Project, which is a wonderful organization founded by Peter Neufeld and Barry Scheck, And Peter actually wrote a very nice blurb for my book.
Mhm.
But anyway, they've done well over five, maybe by now 600 DNA exonerations of people who are wrongfully convicted, and many of them were actually on death row. So I think DNA analysis with PCR has really transformed our criminal justice system.
It's such an amazing thing because, as we get out of the archaic fingerprinting, kind of technology, to be able to get a genome from everybody who goes to the criminal system, criminal justice system, I think it's important for, the work that you're also doing, in addition to finding DNA evidence, just having that genome on file so that people can go and follow up, for future cases of possibility. Is there any way that PCR evidence is challenged in the courts to be less than accurate? Like, a case like the OJ case comes to mind.
Yeah.
There was obviously a lot of evidence there. There was a glove that, that didn't fit, kind of thing, but there was evidence there. Why was it not utilized to be able to, say, hey, this is your blood, it's here, and
Yeah. Well, yeah, let me answer that in two parts because they're really two different questions. When we first started doing tests and this HLA DQ alpha test with PCR, that was the first forensic test kit that was distributed. And so my early experience in the criminal justice system was not terribly edifying. That, if the DNA matched, then the defense would argue that it was garbage. And if it was an exclusion, if the DNA didn't match, then the prosecution would say, oh, this is not reliable. So, and that was emotionally kind of difficult because I testify in cases and they would basically try to trash you. Isn't it true, Dr. Erlich, that that's just a bald-faced lie? Someone actually asked me that. But over the next few years, it was established that it was totally reliable. But you're right, in the very first cases, because we have an adversarial justice system, it was always challenged.
Mm.
And one of the challenges was the potential for contamination. That if your system is so sensitive that you can amplify and detect a single molecule, there is the potential for contamination. But there are things you can do to make sure that your system isn't contaminated. You can always run control experiments to demonstrate that there's no contamination. But also, sometimes people would argue against admitting the DNA. In admissibility hearing, the judge decides whether the evidence is sufficiently reliable to be presented to the jury. So a lot of times I was testifying in admissibility hearings. And they would raise this issue of contamination. But, over time, it became pretty clear that it was reliable. And it's interesting you should mention the O.J. Simpson case. I have a whole chapter in the book. The book is Genetic Reconstruction of the Past. If I can shamelessly flog it.
No, please. That's why I brought that up, because I saw the blurb in your book. So please do that.
So I had a chapter on the O.J. Simpson case, and I was actually scheduled to testify in an admissibility hearing. So I got a chance to see all the evidence, and it was pretty overwhelmingly, it was convincing. But in this particular case, Peter Neufeld and Barry Scheck, who, as I mentioned, are kind of heroes of mine and friends, they founded the Innocence Project, which has, I think, been enormously important. They were on the defense team. And so they ended up actually stipulating that DNA per se is reliable, and so there's no need for an admissibility hearing. But then they went on to argue that the LAPD screwed,
Tampered.
with chain of custody.
Tamper with evidence.
Yeah, we all remember Mark Furman
Yeah.
and the implication that may have planted something. But in my view, the DNA evidence was overwhelmingly clear. But still, I had a friend who was on the defense team, very knowledgeable friend, and his view is, the judgment shouldn't be about whether he did it or not. It should be about whether the prosecution made the case. And this friend, Ed Blake, who I work with on a number of cases, he thought they didn't make the case. And my view as a research scientist, not as a lawyer or someone involved in the criminal justice system, I naively think it's about the truth. He either did it or didn't. And maybe the prosecution didn't make the greatest case. Like, as you mentioned, the famous if the glove doesn't fit.
Yeah.
Well, what happened was the glove had dried blood on it and was hardened.
Yeah.
So of course it wouldn't fit. It wasn't a question. I mean, it was his glove. So if it hadn't been hardened with blood, it would have fit. But because it was hardened with blood, it didn't fit. And so that became a big part of the court proceedings. Is, to me, total red herring, totally irrelevant. But
100%. Yeah, it just it became showmanship, and
yeah,
that's kind of what happens in a lot of these cases, that the evidence itself gets lost because it becomes this whole politicized showmanship kind of case, and when you have enough money, I guess, and fame, then being guilty or innocent may be less important than, as you said, who presents a better case. And
yeah, no, OJ was able to assemble a very impressive team of lawyers.
yeah, for sure. I wanted to ask you some more on PCR and what you think sort of the future advancements on personalized health and wellness and genetic testing. Do you think that PCR is part of the future of, precision? Like my company does precision health and wellness genetic testing and analysis. We're using sequencing. Do you think that PCR, and the reason why I bring this up, because one of the biggest challenges that we have, with our customer base is they want the results immediately. They want it now. And it doesn't, what we do, it doesn't work, and they don't understand. It doesn't work that way. So is there a way to accelerate using PCR technology, maybe for precision health and wellness?
Well, by now I think PCR is just a part. At one time, it was a hugely important technology in molecular biology. Now it's just part of the background almost. It's incorporated into everything, but it's kind of a foundation, and other things are built on that foundation. So as next generation sequencing is enormously powerful. Um, we do it in some of the studies we do. We're now actually trying to do prenatal testing using fetal DNA in the mother's blood, so it's non-invasive. And we use what's called next generation DNA sequencing.
Right.
But next generation DNA sequencing is, as you know, a strategy with a number of different platforms that can achieve this. But you have hundreds of thousands of parallel DNA sequencing reactions, and they all start from a single DNA molecule. So it's an ideal technology for analyzing mixtures, because, like fetal DNA in the mother's blood, because each reaction is the individual component. So you're sequencing the individual components of a mixture separately. But as you also know, it's very powerful. You can sequence the human genome in several days, whereas it used to take millions of dollars and months and months and months to do that. And also, some DNA sequencing technologies take a few days, but we're also using one that's very fast. You may be familiar with it, Oxford Nanopore,
Mhm.
which is a very interesting technology, and you can get a sequence in an hour or two. So, that might be useful for some of the work you're doing. But, we use other next generation DNA sequencing technologies that take more time, but there are additional benefits to those. But anyway, so it is possible, I think, in the future that getting DNA sequencing results will be faster, and PCR will continue to be part of that. But it's like the first brick, and there's a whole edifice based on that.
Yeah, it's first generation too, and that's why I wanted to ask you a little bit about, AI, how that's gonna, because we already have like low pass sequencing where there's, you can use technology instead of doing whole genome to
Mhm.
be able to predict what the outcome may be. How do you see the next or the future of technology with all this machine learning? How do you see that sort of accelerating the genetic work that you're doing, your colleagues are doing, in the future of this personalized health and wellness?
Well, I think it'll be able to provide correlations that without machine learning or AI, we're not able to do with our simple human brains. That all these molecular biology technologies like next generation sequencing are going to generate huge amounts of data. And there are all kinds of bioinformaticians who are analyzing it, but I think the analysis will be aided significantly by machine learning and AI. The pattern recognition that AI is capable of is much more sophisticated and powerful than anything that the human brain can do.
Yeah, no, exactly. What I agree with you. What prompted you to write a book, and what was the process of writing your book?
Okay, now it's a good question because the book is about two seemingly disparate fields, but actually have a common thread. So the beginning of the book is how we develop PCR. And I talk a lot about, I try to give the flavor of doing science, doing research, and how the scientific enterprise differs from a lot of other scholarly work. So, I emphasize that it's a team sport. I emphasize that in addition to creativity and innovation, which everybody appreciates about science, skepticism is a really critical element in science. And I talk about control experiments, how you're always checking to see is if what you think is going on really is going on. And you always have a negative control and a positive control. You're always, in a way, checking that your hypothesis is correct. I mean, one of, there's a philosopher of science, Karl Popper, who says that science is really about creating hypotheses and trying to falsify them.
Right.
Um, so I talk a little bit about science in general and a tiny bit about my own personal journey, how I got interested in genetics and biochemistry in college. And then I did a postdoc, two postdocs, the second one at Stanford in immunogenetics. And how I took a job at a little biotech company in the Bay Area. And the reason I did that is because when I was at Stanford, my girlfriend, who was a professor at Oberlin College, moved out to San Francisco with her dance troupe. So they all came out in a big yellow school bus, her three kids, her 10 dancers. And so since she'd kind of moved everything to San Francisco for me, I needed to find a job in the Bay Area. And I took a job with this little biotech company. And I'd always been inclined to, look at universities, but here was this biotech company, and the advisors were, professors I worked with at Stanford. So I thought, how bad could this be? This could be fun, and it was. And as I put in the preface in the book, the culture was more counter than corporate. It was like a bunch of hippies trying to figure out things. So it was very exciting to work at CETUS. And that's where we developed PCR. And then so the book goes on to describe how we amplified beta globin, but
Yeah.
then how we amplified this polymorphic HLA gene and started to do forensics. And so my interest in writing the book, and the title of the book kind of gives away the concept. It's called Genetic Reconstruction of the Past, DNA Analysis in Forensics and Evolutionary Studies. So
Human evolution, I think, right?
yeah, and so they seem like two very different fields, two disparate areas of research, but they're both about the past. One is reconstructing the crime scene, the recent past, and the other is trying to recreate the human evolutionary past. And so, part one is all about my experience in the criminal justice system, and I talk about the O.J. Simpson case, all the exonerations that I worked on, how DNA databases are screened, how the metrics used in forensics are calculated, what assumptions are made. Like if a DNA profile of the evidence matches a suspect, what does that even mean? And the metric that's used to interpret the significance is called random match probability. And what that means is, what is the probability that someone picked at random in that population would also match? So in the beginning, when we had just a few genes, the random match probability, might be 1 in 500, 1 in 1,000. Now, as you know, with lots of different genetic markers that are used, it can be 1 in 100 million, it can be 1 in a billion. So I try to explain all those things in part one. And in part two, I talk about the work done on ancient DNA. I talk about the work of Alan Wilson, who was a pioneer in molecular evolution and was a professor at University of California at Berkeley when I was at CETUS, and we sort of taught his lab how to do PCR. And he, had the idea to use PCR to try to amplify DNA from extinct species and ancient DNA. And one of his postdocs, Svante Pabo, who was at Berkeley at the time, and now is the former director of an institute in Leipzig in Germany, he also got the Nobel Prize a couple of years ago for sequencing the Neanderthal genome.
Yeah.
So in the book, I talk about ancient DNA and human migrations and what stories DNA can tell us, and how we know that, what are the assumptions we make to try to interpret the data. And then there's also, a chapter about genetics and race, and how, race is really a social construct, not a biological or genetic one.
I'm so glad. I wasn't even thinking of like asking this question, but since you brought it up, it's interesting because I've done my whole genome. I've done all these different. I did my 23andMe. I did our own. I did a but in terms of race and ethnicity, I'm 98% Ashkenazi Jewish, 2% Scandinavian, which I don't know where the hell that comes from, but maybe it's there. I do have blonde and blue-eyed people in my family, but I always found it fascinating what you just said, being Jewish, and and I'm not going to talk about politics and all the stuff that's going on in the world. Unless you want to go into that, but I always found it fascinating that people have a really hard time understanding the religion of being Jewish versus the ethnicity or the genealogy that's associated with that. And I'm like, I do my DNA test. It actually says, my ethnicity, whether I practice, I can be a Buddhist. It doesn't matter what my religion is, but I'm trying to get people to understand, and they have a hard time with that. Is there a way to explain that to people better than how I've attempted to do that?
Well, I mean, it's interesting that I think I have you beat by 1 percentage point. I'm 99% Ashkenazi Jewish. But so my wife was much more interesting. You know, she was like 17% Jewish and everything else, a lot of other different things. But I think one of the important things for people to understand about ancestry and genetics is that every group is incredibly genetically diverse. So the idea of race or ethnicity as homogeneous
Right.
is not case is not the case. That when we try to use these ancestry algorithms, we're actually focusing on that tiny part of the genome that is informative. So they're, as you may know, they're called AIMs, or ancestry informative markers. But most of the genes, well, first of all, 99% of all our genomes are identical, and less than 1% varies. And there's much more variability or diversity within a population than between populations. And that's really why the idea of a homogeneous racial or ethnic group is it doesn't make sense, doesn't fit the data. But as you say, if you use these ancestry informative markers, which are just a tiny portion of our genetic variation, they do allow you to categorize people into groups. So you and I probably have a huge amount of genetic differences, but we're both 98, 99% Ashkenazi Jewish. As you point out, it's an ethnicity, it's not a religion. You know, my parents are not religious, I'm not religious. Um, but nonetheless, that is an ethnicity.
Yeah, it's a great way to explain that to people because I was trying to, have these conversations like, no, it's religion. I'm like, I mean, yeah, so what? But you're not 99% Buddhist. You know what I'm saying? It's not
Yeah, Buddhism is not an ethnicity, it's a
right.
religion. Yeah.
Exactly. I wanted to ask you about sort of a two-part question. Your interest and your continued interest in genetics, do you still have this interest and drive in forensics and the future of the criminal justice system? Or do you have sort of this passion for let's solve disease states and seeing how we can address, or is it a combination of both?
Well, I am very interested in both areas, and even though I would say semi-retired, I'm emeritus. You know, I'm 81, so but I still have an office and a lab, and I go in and chat with people there. I don't direct, I give them guidance and advice, but I'm not, and I might write the paper, but I'm not, bossing people around in the lab
Anymore.
Anymore. Actually, to be honest, I probably never did that, um, but yeah, no, I'm interested in both areas. I kind of do a lot of reading in both areas. The one thing in the lab, I did have a grant from the National Institute of Justice to use next generation DNA sequencing to look at forensic mixtures. But I also had a grant that I'd been working on, and I'm actually still working on that one, to do non-invasive non-invasive prenatal testing. That is, look at fetal DNA in the mother's blood, so there's no risk to the fetus. And I'm looking at what are called the beta hemoglobinopathy, sickle cell anemia, and beta thalassemia.
Right.
And to be honest, I think I just enjoy the intellectual challenge of trying to figure things out.
Yeah, it's a better than doing a crossword puzzle.
Oh yeah, for me, much better. It's funny. At one time, when I was a kid, I was very interested in chess, and then I decided, no, for games, I'm going to play basketball. For intellect, I'm going to think about science.
Yeah, I love that. Wanted to ask you about your thoughts on plant medicine.
Plant medicine.
Plant medicine, yeah, because one of the things I think you mentioned previously that there was a polymer, something that was discovered from like
Oh, Yeah, the DNA polymerase
That's it. That's it.
isolated from the bacterium Thermus aquaticus,
Exactly.
which is this is funny. This is a bacterium isolated from the hot springs in Yellowstone National Park. And my brother was there, as a tourist, and he said that's now part of the park ranger spiel. They talk about how this enzyme was isolated from bacterium in its hot springs.
I love that. So I guess I'm trying to. So the question about plant medicine is, you know, there's a lot of there's medicine that we synthesize in a lab and we create that, but a lot of it came from plants in the first place.
Yeah.
And we have the Amazon that's untapped. There's so many resources there. There's cannabis, there's psilocybin, there's all these different things that are, grow in nature. And if, one of the things that, my company has a patent on a DNA test associated with the endocannabinoid system, but there's also, exogenous delta-9 THC that will bind to those receptors and release your anandamide and 2-AG levels and all that stuff. So there is plant medicine. We can actually track to see that consuming the plant makes changes in genetic expression as well. So I just want to get your thoughts on plant medicine, if that's an area that you think for the future of next generation, if we're going to start incorporating a lot more of that plant medicine.
Well, I don't know much about it, but I think it's a really fascinating area with a great future. I mean, many of the most important medicines have been isolated from either plants or microorganisms. Like, of course, penicillin was from molds.
Right.
And I think we've not tapped the potential at all yet. So I think that would be a very exciting area to work in.
Well, that's if in your semi-retirement, if you're bored, let's definitely talk about that kind of stuff. I'm fascinated. It's one of the things that's driving me in the research that we're doing with our company. I wanted to ask you sort of your thoughts on the next, maybe 10 years in genetics. What do you think is going to be exciting, and what particularly excites you in the future of genetic research?
Well, I think one of the really exciting areas for me is ancient DNA and what it tells us about our history. And I think we're constantly learning more and more. There's more and more work being done. It's a very vibrant field in molecular biology. So that's certainly one area that I think will be transformed, and it's being transformed by the new technology. So, a few years, let's say 5 or 10 years ago, it was very challenging to try to sequence the genome of an ancient, of a fossil like a Neanderthal skeleton. Now there's several complete genomes from Neanderthal samples. And I don't know if you're familiar with this. I do talk about this in the book. There's an entirely new as of about 5 or 6 years ago, archaic human species called the Denisovans. Are you familiar with that? Have you heard? So anyway, we all know about Neanderthals.
Yeah.
But in a cave in Denisova, Siberia, there was a Neanderthal skeleton, but there were also some other bones. And there was a tiny, tiny finger bone that the scientists were able to sequence. So tiny finger bone, they got both mitochondrial DNA sequence, which is much easier to obtain because cells have thousands and thousands of copies of the mitochondrial genome, but only 2 copies of the nuclear genome. But anyway, they got mitochondrial DNA and they got nuclear DNA sequenced. And it wasn't modern human, it wasn't Neanderthal. So it defined a new archaic species that we had heretofore been totally unaware of, and now called Denisovans. And now we also know that modern humans interbred with Neanderthals, and about 2 or 3% of our genomes are Neanderthal. but Denisovans also interacted with modern humans, mainly in Asia and Southeast Asia. So, people in Indonesia, Papua New Guinea, Australia, other parts of Southeast Asia have 2 or 3% Denisovan DNA in their genomes. So that's one of the most amazing things that has emerged from, the application of molecular biology to human evolution. And it's largely technology driven. Um, it's modifying the protocols for next generation DNA sequencing to be able to analyze very small DNA fragments, because these really ancient DNA samples, the DNA is really in tiny, tiny little bits. So the technology to sequence and assemble a genome is what's enabled us to look at that.
Yeah, I love the study of, the history and because it tends to repeat itself also.
Yeah.
So understanding, what happened in the past can help us, maybe predict or take action against the expression in the future. I have a few questions I ask all my guests, not genetic related or anything like that. More music stuff, because I'm interested in that. Do you remember, and if so, what was the very first concert that you ever attended?
I'm trying to think. It might have been either Ray Charles or James Brown when I was a teenager in Seattle, growing up in Seattle. So those are two of my heroes.
Yeah.
And I've seen both of them many times. But I also like classical music. I actually played in the Seattle Youth Symphony for a while, clarinet. And when I was a teenager, I also studied classical guitar a little bit.
Yeah.
But so I continue to really enjoy music. Actually, I just went to a bluegrass concert the other day, and we're very lucky in the Bay Area. There's a club in Berkeley called Freight and Salvage, and I heard a singer named Liz Wright. I don't know if you know her work, but
my daughter actually goes to Berkeley, so I was just up there a few weeks ago for Parents Weekend, and today or this weekend is the big game against Stanford.
Yeah.
So they're getting prepared for that too.
So Len, where are you based?
I'm in LA.
You're in LA, yeah. So my kids went to Berkeley. My grandson just graduated from there a couple of years ago, actually. It's a great place, Berkeley.
Fantastic school, like the whole
Yeah.
thing. I love it, except for the protests and the sit-ins and all that other stuff. The encampments. Maybe I'll start cleaning that up a little bit now. So another music question. For next year, you can only listen to 5 albums, and you don't have to remember the name of the album, but you can just say whatever the artist is. What will be those 5 albums for artists?
And they can be passed away.
Oh yeah, for sure.
Oh yeah. Okay. So I have pretty eclectic taste. Ray Charles is one of my heroes, but Merle Haggard is, I've seen him many times. So I guess it'd be Ray Charles, Merle Haggard, and then maybe the Magic Flute, Mozart's Magic Flute. I just saw a production of the Magic Flute at the Berkeley Rep about a week ago, actually. So yeah, Mozart, Ray Charles, Merle Haggard, and then maybe the Modern Jazz Quartet. I've always really liked them. So that's 4. I got 1 left.
Yeah.
Maybe something by Bach.
Okay.
A fugue, or
Yeah, I'm surprised you didn't put James Brown in there, since that was one of your. Yeah.
Well, no, I really like James Brown, but Ray Charles for me it was a different level.
Oh, for sure. James Brown would probably say that if he was alive too. All right. So, final bonus question. Please describe what your room looked like growing up.
No, that's pretty funny. Okay, I think it was painted blue. I painted it blue. I had a Van Gogh print on it. And the window looked out on the backyard, growing up in Seattle. And had a radio. I used to listen to the radio a lot, music and various shows. Yeah, that's kind of what I remember.
No artwork on the walls, no.
Well, no, there was Van Gogh print.
Yeah, besides that, yeah, you said that.
Yeah, I can't. That's actually, that's interesting you mentioned that. That's the one thing on the wall that I remember. And, when I was, say, 10 years old, so that's over 70 years ago. So
Right. Which Van Gogh print do you remember?
it was his room.
Oh,
His room in Arles. There was his bed and
very interesting. Because I used to have the sunflower
yeah.
print on my wall when I was
Yeah.
a kid, interestingly enough. Henry, where can people find out more about you, where they can buy the book or anything else that you want to share with our audience?
Well, the book is Oxford University Press.
Mhm.
It's called Genetic Reconstruction of the Past, DNA Analysis in Forensics and Human Evolution. And I'm not that much in the online world, but my daughter is convincing me to get a website. So she's got someone building a website for me. I'd give it to you, but I don't remember
That's
what
fine.
it is.
People can Google you. There's plenty
Yeah,
of
yeah. No, that's right. If they Google me, eventually the website will be on there, I guess.
And for my audience, I hope that whoever stayed with it is really interested in genetics. So I appreciate everybody listening, and I really appreciate everybody's reviews and feedback. It's been fantastic. It makes me learn things that I can improve on, and just the impact that the show is having in people's lives. I really, really appreciate it. And thank you for buying my book, Making Cannabis Personal. And, Henry, I so appreciate your time. I mean, what a contribution you have made in the world of science and genetics. And I don't even think that people realize the impact of what your discoveries, what your findings, and what the future of the work that you're doing now, what you're interested in. So I really, really thank you for your time and your contribution. Appreciate it.
Well, yeah, thanks a lot, Len. No, it's been a real pleasure.
Likewise.
Yeah, take care.
You too.
Bye.
Hey everyone, thank you for watching and listening. And don't forget to hit the subscribe button, share this episode, and hit that bell. Like, comment. I so appreciate that. And check me out on social under Len May or Len May DNA. Find out more on LinkedIn or lenmaycoaching.com. And remember, everything is personal.